Furthermore, Richard is a member of the ISO TC 198WG9 Aseptic Isolator Group. Richard was on the Annex 1 and the PIC's Annex 2 commenting team of the ISPE. Richard founded 12 years ago the Containment Expert Group of the ISPE DACH. The Containment Group published the Containment Manual. Richard was responsible for that in September 2015. Richard has spent more than 20 years with the subject on aseptic processing and highly active, highly hazardous substances and has developed the containment permit. Welcome to Dr. Richard. The floor is yours.
Okay, thank you, Dr. Chika. Can you see my screen?
Yes.
Okay, so also a warm welcome from my side. My talk will be, or let's say good morning, good afternoon, good evening. It's a global virtual meeting, and so you never know, and don't know from all over the world that people are joining this seminar. So I will give you a little bit a different presentation of what I have seen on the overall program. I will talk in my 40 minutes more about manufacturing of cell and gene therapies, and there which guidelines are required, and will give you also some details what has to be considered. So as already mentioned in my biography, I'm a mechanical engineer, work for and started also a GMP many years back. I work for a company, for SKAN AG, based in Switzerland. We are a world leader in aseptic processing for manufacturing, let's say, sterile products. There I have mentioned a few positions I have globally, maybe to explain that to those who are not familiar. The ISPE is an international association for pharmaceutical engineering. It's a non-for-profit organization, provide guidance to the industry. And there is another one on PDA. This is Parenteral Drug Association, also a global non-for-profit organization focusing on quality control. So why is this manufacturing part hopefully also of interest for those attending? You know, we expect a lot of new therapies coming to the market in the next decades. Many of those will be in cell and gene therapy or tissue engineering. And from their clinical studies, let's say it like this, or they're coming from university or from big pharma, they have also to go in commercial manufacturing. And this is where I fill the gap. You know, I take a lot care about guidance, documents, how to manufacture, GMP compliance, and how this can be applied also for new therapies. And that I'm very well known globally, and I do give a lot of presentation about this topic. So in the beginning, I will talk a little bit about GMP requirements, because it's that what you have to apply when you deliver therapies to the patient, and followed then with how to facilitate the design of what is there important to consider. So in general, there is more requirements than those two, but on those two I'd like to focus in my presentation. There is PIC's Annex 2A. So it's PICS means Pharmaceutical Inspection Cooperation Scheme. This is a group of 52 member states where they make guidance documents for inspection. And there is a new guideline published last year about 2A for ATMPS, for advanced therapies of medical products. There I will go a little bit in detail in the beginning. I was member of the ISP commenting group on this document. And there is another document also this year published, was Annex 1, which is also for PICS Annex 1, but this Annex 1 is for the EU GMP guidelines, manufacturing sterile products. There was also a member of the commenting group. So Why is this important for you? So this PICS Annex 2A is a regulation which our 52 member states belongs to. And this is focusing on ATMPs like cell and gene therapies. And with Annex 1 for sterile products, that means even when some of the products are not considered as sterile when they come from the patient, you should avoid any contamination to the cells when you receive them. And for this, in the PICS Annex 2A for ATMPs, they refer also to the PICS Annex 1, which is the same as the EU GMP Annex 1. So on the points to consider document, what is mentioned below for the manufacturing of ATMPs, this is under development, but I'm there a chair of this working group, and there we have a global team from Japan to Brazil, from North America to Australia, Europe, all over the world, where we work on requirements for manufacturing of these new novel therapies. So PICS Annex 2A, as mentioned, is published since 2 years, and I was there involved in the commenting team because I met the head of this PICS Annex 2A working group in Brisbane, Australia. He was at that time working for the Therapeutics Good Administration based in Australia for the Australian market, and there was also the involvement in this Annex 1. And he had some questions on the revision of the document where we were then discussing and did a lot of communications. Okay, this was quite an interesting exchange of between the industry and the regulatory bodies, which did not happen in that way before. So there were 8 specific questions sent out, where we from the expert group of the ISP commented back. And I will only highlight the 2 of those questions. One question was quite important: when does GMP start and what other materials should be included in the table? Because in the PICS Annex 2A, there is a table, which you can see on the left side, where the gray part is that what is mentioned in the guideline, where the GMP requirements are applied to. This is there mentioned like for vector manufacturing, cell isolation, and so on. And then was another question out of the 8 was, because this is something what all of the regulators have big concerns, because when you manufacture not in, let's say, an appropriate technology, like in closed systems, then you can always spread the material in other areas, which can create a contamination of the operators working in that field, or a potential cross-contamination to other patients. This is quite important to consider, and this you can also find in the PICS Annex 2A, that there is high concerns when you work with cells from the patient, which they can be already toxinated, and they work then often with viral vectors, which are programmed viruses, and when there is some contamination or cross-contamination with other treatment of the patient possible, that can be fatal effects for the patient. And there was in the question mentioned, what are your views on the expectation for the working environment requirements when processing is not performed in a closed system? When you look at this, they already require here closed system. The same I will come then to the next slide when we talk about the EU GMP Annex 1. And there we have then commented back, because contamination, cross-contamination is a high concern when those new therapies go into the commercial manufacturing. So this is the 2 points what you can what I wanted to highlight out of this PICS Annex 2A guideline: contamination of product from the surroundings. So you should avoid working with material which you see from the patient, that this is contaminated microbial or particulate from the surrounding, or a cross-contamination and product mix-ups during manufacturing. So product mix-ups is often in cell and gene, as I mentioned, really a fatal effect when you would mix up from patients different cells, because patient can have highly toxinated, as I mentioned, they can have HEP, they can have other diseases, and when you have there a mix-up or you cross-contaminate with other product can have their fatal effects for the other patient. So GMP Annex 1 requirements. This was also a document I was involved with the ISP on the commenting of this document. This was a little bit differently done because there was not face-to-face meetings. This was more that the European Commission sent out questions to the stakeholders like ISP and PDA or others, and we could then provide feedback on it. So, and this is a document where the journey started already 2015, where the first not more internal of the guidance group provided the first draft. In 2017, the first draft was sent out public to the industry, where the industry could give feedback on it, and they received a lot of feedbacks. Then was in 2020 another feedback sent out the next version, and then in August 2022 was then the publication finally of the document, and there is an implementation time of around 1 year. And there is something also quite important mentioned. In principle, I will read that because this you will see then how it goes through the document. Facility, equipment, and process should be appropriately designed, qualified, or validated, and where applicable, objected to ongoing verification according to the relevant section of the Good Manufacturing Practice Guideline. So the use of appropriate technologies like restricted access barrier system RAPs or isolators, or also robotic system, rapid alternative method, and continuous viable monitoring or monitoring should be considered to increase the protection of the product from potential external sources of endotoxin, purogene, particulate or microbial contamination, such as personal materials and surrounding environment. So there's it's now clear mention that you should avoid that from personal contamination should come to the product. And there we have seen specifically in that area a big shift in the last 15 years. So when I look about manufacturing for cell and gene, it remembers me many years back in my career where we had a similar situation. So the left picture shows you similar operation in aseptic operation for sterile product. This is where you, for instance, fills vaccines in vials or syringes. And this was mainly done in an open process. As you can see here on the left side, there were people dressed in clean room glass B, for instance, where here open containers were running in that area, sometimes protected with curtains. People are gowned, but as you can see here on the operator left, on the right hand, very close to open sterile containers. And this, as you know, humans are the highest risk of a potential contamination or also cross-contamination when they contaminate themselves. This is now almost gone in that specific industry. In the meantime, only closed system like isolators, as you can see in the right side, are used, and they go even forward to fully automated system where no operator has access to the inside with gloves, where all of the operation is done with robotic system. So we are quite far away in the field on a cell and gene therapy or for new novel therapies, but the guidelines are driving in that direction. And we will see there also a lot of shift into more appropriate technologies in the future, which I explain to you in the next part of my presentation. But also Annex 1, there is a contamination, there's the concern, the contamination of the product from the surrounding, cross-contamination, product mix-up during manufacturing, the same as mentioned in the PIC Annex 2A for cell and gene or ATMPS. And here, additionally, in Annex 1, contamination control strategy, what you have to apply when you manufacture those products. So you have to provide a strategy where you can demonstrate that during manufacturing the product is protected. So both Annex 1 and PIC Annex 2A have their preferred closed system, which I will explain you in the next part of my presentation. So there is something, it becomes now a little bit technical. I have seen on the 2 days, there is a lot of scientist presentation, but in the end, you know, whatever you develop, wherever you work on it, it has to be manufactured. Otherwise it cannot be applied. So this is that where the most companies fail right now. They develop something without thinking about how this can be developed. But when it can go in commercialization, then they fail, that they cannot be processed that it is GMP compliant. And for that reason, you know, is it quite important, even in early beginning of your development of the product, to think about how you can process for commercial manufacturing if you would not to fail then with a very good therapy, which then is not possible to manufacture GMP compliant. So what I wanted to tell you is, in general, you have to generate a GMP environment, which means you manufacture with your process in an environment which have normally a primary and secondary containment control. Primary containment is in general something what you can see here, this biosafety cabinet, which you can install in a grade A, B environment, or you can go with a more advanced technology, which is that one on the right, which is Isolator system, which is mentioned in Annex 1. Because there is a clear mention Annex 1 that you have to provide a barrier from the outside to the inside. And when you look about the left picture, there is a gap here below the window where the operator can have access all the time to critical operations inside. So this is not considered as a barrier system. And for instance, when this PIC Annex 2A for ATMPS is linked to PIC Annex 1, which is also refers to barrier system. Such kind of biosafety net cabinet system is not likely to be used, as it's the highest risk of contamination of the product and also of the surrounding. So this is your primary containment that you should try to isolate your process and your GM manufacturing inside of the process as close as possible. And your secondary containment is in general the room outside. And the room outside consists in general in materials and personal airlocks, have then the working area. And when you go in, when you look into new Annex 1, you should have unidirectional airflow. That means preferable is also that you have material and personal flow in. You have your manufacturing process in the middle, and then you leave also unidirectional this material and personnel out of the area. It has also advantages, and there we have in this points to consider document, what I mentioned before, where I'm the chair with a global team, and we hopefully that we can publish this by the end of the year. There we have also examples in it, you know. What does this in general mean when you have clean room classification? So there we have, when I compare it to European GMP classes, A, B, C, D, and then is that what we see here in the different colors. And there is 2 examples. There is one example when you go with biosafety cabinet in the grade B environment, where you have the grade A airflow inside of the biosafety cabinet, or when you use isolators on the right side. Yeah? There's a huge impact because when you have a lower D grade, as you can use for isolators, means less gowning for the people, less monitoring, less air changes, less room. Yeah, you can see the space needed is in general 30 to 30% less, and the operators have not to be intensively trained and gowned working in that area. So you can also much easily have the air flow in GMP compliance installed, like say that you always avoid that you have airflow inside of the critical air, in the critical area where you process. For that reason, you want the airflow, which is there centralized when you work specifically with viral vectors, with viruses, that they do not spread outside, yeah, and could contaminate the surrounding areas. As you all know, since we are exposed to COVID, we know how easily how we can contaminate it with viruses, and in the meantime, operators in that areas are very sensitive working with that. So, generally, I wanted to give you a few information about the pros and cons of such kind of different system. In general, the investment costs of such biosafety cabinets are really low here compared to an isolator system. This is why they often are used from the companies, but in the end, you know, they have the concerns that the operator gets all the time in contact with aseptic sterile operation. It's not considered as a barrier. So when you go in commercial manufacturing, you might have troubles to justify this technology. You can have a contamination from outside to the inside and also from inside to the outside. So people can be exposed to your product, what you like to handle inside. And the higher demands get also in the secondary containment as room, as many of those cell and gene therapies are in biosafety level 2. That means you have to have protection also that nothing of the material is spreading outside of the system. Actually, what I face on what we investigated, every second new therapy in that field have been considered in biosafety level 1 or 2. So what there is also a choice to use a single-use closed system. This is also something very popular to use. You have flexible bags where you process all of your manufacturing step inside. The only thing what you have to take in consideration sometimes is very complex, the assembly and also the design of the connectors have to be in sterile way. When the design of the connectors cannot be considered as sterile, then you also have to make it inside of an isolator or a closed system. So when we think about the isolators, what I mentioned before, which you have the highest protection of the surrounding, as you have validated airlocks. I come to this a little bit later on it, where you transfer material in. You have a closed system where you have only gloves from outside for manipulating inside. Here in that application, you have an integrated centrifuge, and a centrifuge is here on the bottom and an incubator here on the side that you can process your whole manufacturing batch without leaving that area. So the cons is of this is that you have much higher investment cost of the isolator, but this compensates on already after several years on the lower secondary containment requirements. And the operator have to work through a barrier. He has not full access to it, but it also protects him. Yeah. So the operators are also protected during manufacturing. And what is often the case, fast transfer inside. Yeah. People or companies require fast transfer of the material. And this is also something, but there is technologies on the market where you can transfer in minutes now material inside with such hydrogen peroxide airlocks. This is such a little bit about comparison. What is there, the pros and cons between biosafety cabinet and isolators, where they are more beneficial, where the advantages are higher and the disadvantage lower, and also the other way around. And in general, you can face that even when the investment costs are higher, there you save and give much higher protection to the product. So when it comes now to higher biosafety level, this is something where we face in the meantime, there is no way beside doing this in a closed system like isolators, because in that moment when you handle inside a closed or open materials and work with viral vectors, the exposure risk to the outside, excuse me, the outside and to the other areas is too high. And there you have the advantage of an isolator system after you finished your batch, you can run an inactivation cycle with hydrogen peroxide afterwards to inactivate all remaining materials inside before you start cleaning and prepare the system for the next batch to be processed. So I will go you a little bit into aseptic processing and a little bit more in the field of isolators. Yeah, why this is there important and why you have then the chance to fulfill in that way the better GMP compliance. So I will talk a little bit about equipment design and also cleanability of the system and surface decontamination. So I have here some information always out of the new Annex 1. You know, it's now published. There's also PIC's Annex 1, which have the same content as the EU GMP Annex 1. And for all of those who are referring to the PIC's guideline documents, there you should also focus on the EU GMP Annex 1, because in PIC's Annex 2, as I said, is the Annex 1 also referred to. And there you can see, when you look at 4.3, this where they talk about a restricted access barrier system, wraps or isolators, which are both closed system, should be used, and their use should be considered in the contamination control strategy. And here it is, any alternative approach to the use of wraps or isolators should be justified. So whatever else you use here, you have to justify this specifically on manufacturing. So what is there important? You have to demonstrate the unidirectional airflow. That's what you can see here with smoke studies in the lower picture below. There you see also such a cell and gene based isolator. And there you have to demonstrate that you provide the first airflow over a critical sterile operation. This is also something what makes it in the future a little bit challenging how to work when there is a lot of intensive manual work to be done inside, because this first air principle you have to demonstrate also with the smoke studies. So the airspeed inside of such an isolator system, what you can see here, is in general also provided between 0.36 and 0.54 meters per second. Here you have this section on the side. How does it normally works? You know, you recirculate the air. That means you have here the terminal HEPA filter. You get through inside the isolator chamber, which is here in orange, and then to recirculate during double door windows the air back on top, and then you get the same, or if then you get the air to the HEPA filter through again and into the critical zone to provide always this unidirectional airflow. Material transfers is also something of concern. To say you should use there also appropriate validated methods. So for instance, when you hand over from outside via biosafety cabinet inside the area where you operate, this is not a validated system, yeah, because it's manually done. A validated system are in general such airlocks. What you can see here, there is one on the left side where you put your items in, what you need in your isolator. Here you can see the centrifuge and the incubator, what I told you before. And there you get all items in. You have a 6-log reduction, normally done with hydrogen peroxide. Then you transfer material inside in a safe way. This is also such an airlock which works in an extremely fast way, less than 3 minutes, and also with extremely low concentration of hydrogen peroxide, because there's often a question, yeah, hydrogen peroxide can also react to our product. And there is also test procedures available which can demonstrate that is not the case. For instance, when you work with such flask, you can tape this here with aluminum foil, where it is the critical part. You get it into the airlock, you make your decontamination cycle, and then you replace only the lid inside of the isolator to a new one. And this one is then disposed. But this is also a thing of a safety, because when you transfer your flask outside, the inner part can be also contaminated through the surrounding. So gloves is also something what you have to think about it when this is the only access what you have from the outside to the inside. So gloves have to be tested, and before and in the end of the batch or the product campaign, whatever you use, it's clearly mentioned in the Annex 1. And you should also do an visual test and check the glove before you use them. So what does this mean? A visual test of such gloves means you have a glove test device. This is that one what you can see in the middle. It's attached then to the glove, which is installed like here in an Isolator system. Then you blow in air pressure over a certain period of time. In general, it takes around 10 to 15 minutes. And then after the 50 minutes, you make a pressure drop test. This is that one where you then check if the glove is the integrity of the glove and the sleeves in the system. But nevertheless, you have also to look on a visual test on the glove that there is not anything damaged, because this, as I said, is the barrier between outside to inside, and there you have to be trained, have trained people how they can identify this. There's also a lot of documents where I was involved on the market for glove testing. Sterility test, it is also quite important because, and also the viable monitoring, this is something has also changed in the new Annex 1, and for that reason, more appropriate technology is also something what you should use. It's shown here below, it's a sterility test isolator where you perform your sterility test inside as this person is doing it in front of the isolator. Here you can see also that they are specifically for a sterility test, as you can put those also in a not GMP classified area in it for make the test. But quite important here, no growth is in the future allowed. Yeah, so wherever you have a growth and a detection, you have to go in investigations, which could lead that you cannot release your product as quick as it should be. So cleaning is one part what I would also like to address a little bit on it. There is also from the PDA there you can see this logo here, points to consider for aseptic processing of sterile pharmaceutical products in isolators. There you get a lot also on information. So if you're interested on this, you can also reach out to me for further questions. So in the new Annex 1 is also mentioned the cleaning process should be validated to able to remove any residues and debris that would discriminately impact the effectiveness of the disinfection agent use, and also minimize chemical, microbial, and particular contamination of the product during the process and prior to disinfection. So your cleaning should now be validated. This, I think, is a big challenge, how to validate the cleaning in the future, because manual operation is often not seen as an appropriate technology to validate or to justify the cleaning process. For that reason, I have set up an expert group many years, around 5 years back, to get more guidance on that to the industry. And there we published also 2 PDA papers. One is there mentioned on the left side, isolators key to prevent cross-contamination. Why we have done this? Because the validation of the cleanliness is not new. Even in the Annex 1 is it's now more clear mentioned. It was also mentioned already in the EU GMP guideline part 1 in chapter 5.21. Depending on the contamination risk, verification of cleaning of non-product contact surfaces, and monitoring of air within the manufacturing area in order to demonstrate effectiveness of control measures against airborne contamination or contamination mechanical transfer. So it's in general a lot that what you do in your, or when you go to the market later on in the manufacturing part, there's a lot of air handling in that area, and you have a lot of mechanical transfers where you transfer the material inside of maybe isolators or other surrounding. And there is then the question how to clean that. And there we have set up a global team on cleaning, GMP expert, and we had also Rico Schulze, is a former GMP inspector from the German inspectorate. He supported us on the two applications we did, because they said there was not any guidance out there. So why is this important? You know, the cleaning has it's a science for itself, yeah, because the cleaning and the results of the cleaning depends on the material you use. Like here, a stainless steel has a different cleaning property than a polymer, or a or a very rough surface, or a very flexible material like a silicone, and also the distribution of the contaminants. So like here, large droplets are much easier to recover on stainless steel than, let's say, very small droplets, which we often have in that field, as there is a lot of aerosols generated. They settle down on surfaces, and also the softer the material gets, where they settle down. Yeah. So the recovery rate can be really go down to an area where you have still a lot of contamination, but you do not recover those. For that reason, I have worked also last couple of years to develop a technology to validate the cleaning process. This is called here Clean IP. This is a printer where you can print exactly the contamination which is normally occurring during the manufacturing on the surface with some safety factor on it. On the surface, yeah, you print your contaminants, could be also surrogate material, and then you start the cleaning process, and afterwards you can then verify if the operators have cleaned the area as it should be. And this provides the first time a validated method to train your people for validated cleaning within your process. So that gets me to the end of my presentation.